Hello guys, good morning. You are welcome to my YouTube channel, the Explicit Tutorials. Please, as you are watching this video, endeavor to subscribe, like, share, and comment. In today's video, I shall be taking us PBB, which stands for Biology and what? Biotechnology. Then you have the topic called Kingdom Monera. Okay, the organisms found in Kingdom Monera are actually called Monerans. They are called Monerans, okay? The organisms found in Kingdom Monera are called Monerans. Again, these guys, they are what? Prokaryotic. The Monerans are what? Prokaryotic. That is because they do not have a well-defined nucleus. The opposite is what? The eukaryotic. Now, a prokaryotic cell is a cell that does not have a well-defined nucleus. Hence, they do not undergo mitosis because there's no what? Nucleus. They do not undergo what? Mitosis. And secondly, since they don't have a well-defined nucleus, their chromosomes are scattered in their cytoplasm. Take note of that. The chromosomes of the prokaryotes are scattered or are found in which of these? I trust you, neighbor. They'll put option A, nucleus, B, gene, C, chromosome, D. Okay, the chromosome of Monerans are found in nucleus, cytoplasm, maybe they will give you ribosome or mitochondria. Please, it is what? Cytoplasm. Their chromosomes are found scattered in their cytoplasm since they do not have what? A well-defined nucleus. Okay. And secondly, the prokaryotes are known to be the most successful creatures on earth. All right, they are known to be the most successful creatures on earth. One, that is because one, they undergo a rapid type of reproduction called what? A geometric growth. Sorry, they undergo a rapid type of growth called what? Geometric growth. All right, they reproduce very fast. Okay, and such growth is called what? A geometric what? Growth. And again, the prokaryotes, they have extra DNA called what? Plasmids. Take note of this. You are going to see this question. The extra chromosome strand found in the extra DNA strand found in the chromosome of prokaryotes is called what? Plasmids. So please take note of that. I said they don't have a well-defined nucleus, and which means that they lack a membrane-bound organelle. That is, they don't have nuclear membrane, they don't have ribosomes, they don't have Golgi bodies, they do not have what? Endoplasmic reticulum, and even a lysosome. They don't have it. Okay. Some of them have these things I just mentioned. For example, now the mycoplasmas. The mycoplasmas are known to be the smallest creatures on Earth. That they are different from what? The other what, group of bacteria. Now, this is your introduction to this topic, Kingdom Monera. Now, on a broad note, there are two sub-kingdoms. Alright, on a broad note, there are two sub-kingdoms of Monera. You have one sub-kingdom, Archaeobacteriodonta. Bacteriodonta. Okay, is it? Okay, Archaeobacteriobata. Sorry. We have Kingdom Monera, the sub-kingdom Archaeobacteriobanta. Okay, we now also have two sub-kingdom Eubacteriobionta. Alright, so these are the two sub-kingdoms of what? Kingdom Monera. First and first, we are going to be discussing everything regarding the kingdom Archaeobacteriobanta. Okay, Now, in reading certain courses, you don't cram, okay? You take your time to understand the trend or the processes or the mechanism involved in such what topic or course. Now, the Archaeobacteriodonta, the it has a division. That is the Archaeobacteriobionta has a division called what? Division, division, Archaeobacteria. Subkingdom Archaeobacteriobionta, then division Archaeobacteria. Now, these organisms are further classified into different species. Now, under division Archaeobacteria, we have the methanogens. The methanogens are otherwise called methane bacteria. Methane bacteria. Please, you can see the way I'm taking it bit by bit. Under division Archaeobacteria, we have the methanogens. Methanogens are otherwise called what? Methane bacteria. Now, these bacteria are found in swampy, marshy places. All right, they're also found in sediments. They are also found in sediments, and they're also found in the uh, intestinal tracts, intestinal tracts of animals. They're also found in the intestinal tract of what? Animals. Now I said methane, the methanogens, otherwise called methane bacteria, they are found in swampy, marshy places, uh, sediments, even the intestinal tract of animals. Okay. Now these are bacteria that have the ability to produce what? Methane. Owing to the fact that they are found in marshy places or swampy places, this methane is otherwise called what? Marsh gas. Marsh what? Gas. And marsh gas or methane is the principal constituent of what? Cooking gas. It is very, very what? Volatile, and it is not toxic. It is not harmful, and it is what? Highly volatile. Okay. You have another organism under division Actobacteria. We have the uh we have the Cephalobus. The Cephalobus are otherwise called what? Acidophile. Okay, thermoacidophile, please. Thermoacidophile. Thermoacidophiles, okay? The Cephalobus are otherwise called what? Thermoacidophiles. Now, as the name implies, sulfur, thermo, then acid. Now, which means these bacteria are only found in places that have high concentration of what? Salt. Okay, acid. Sorry, they're found in a place where sulfur and acid are what? In abundance, like a place where there's sulfur and what? Acid, acidophiles. They are found in places where there are acids. Sulfolobos, they are found in a place that has what? Or sulfur. Now the third one is the halogens. Okay. Now the halophiles, all right. Now, as the name implies, they are found in places that have a lot of what? Salt. Okay, they are found in a place that has a high concentration of what? Salt. They are found in salty places. So these are the three organisms under division Archaeobacteria. Please take note of that. We have the methanogens, we have the cephalobus or thermoacidophiles, and we have what? The halophiles. They are found in salty environments. Now, since we've analyzed the Archaeobacterial biota, Now the most broad subkingdom is the Eubacteriodonta. Just like I gave us before, subkingdom Eubacteriobiota. Please, methanogens are otherwise called methane bacteria. Okay, so we have kingdom, subkingdom Eubacteriobionta. Now, the word eu, like eu, this eu, it means what? True. They are known as what? The true bacteria. Now, these bacteria have striking features, or they have characteristics that differentiate them from every other bacteria. Let us look at the features now. Now, I said they are called what? True bacteria. What are the features that make them unique? We have one, they undergo a type of asexual reproduction through fission. And I told that they have a geometric type of growth. So they reproduce almost every 20 minutes. They produce almost every 20 minutes, and they produce and grow very, very fast. Okay, that is for that. Two, they 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 lack that is nuclei, the plural form of nucleus. Excuse me. Sorry about that. The kids were making noise, so I have to stop them, okay? Now I said they lack nuclei. The word nuclei is the plural form of what? Nucleus. Now, since they don't have nucleus or nuclei, they do not undergo, there is no mitosis. They do not undergo what? Mitosis, because they lack what? Nucleus. And three, that is the DNA of bacteria. The DNA of these bacteria replicates and distributes itself into two daughter cells. Now, which means that bacteria, they reproduce through what? Replication. Their DNA has the ability to what? Double itself, that is, replicates, distribute itself into what? Two daughter cells, which means that two daughter cells are produced during the replication of the DNA of what? The bacteria. Please take note of that. Now, number 4, like I said, they do not undergo mitosis. But they undergo genetic recombination. They do not undergo mitosis, okay? But they undergo genetic what? Recombination. This recombination is by conjugation, transformation, and what? Transduction. All right. Now, these are the striking features of these guys. That is the kingdom Eubacteriobionta. Okay. Now, of course, this subkingdom Eubacteriobionta has a division called the Eubacteria. Okay. Just as I said when I was explaining the Acteriobionta. Okay. So, asterisk reproduction, they lack nuclei, so no mitosis. DNA replicates and distributes itself into two daughter cells, and they undergo genetic recombination by conjugation, transformation, or transduction. Okay, take note of that, please. Now, under this bacterial biota, we have numerous organisms having different shapes. We have numerous organisms having different what? Shapes. Let me state it here. Now, the bacteria that occur in the form of what? Uh, Bacteria that are spherical or round. Spherical bacteria. Spherical bacteria are called cocci. Singular is what? Coccus. Now, we have bacteria that occur in a spherical manner, and this spherical bacteria now occur in the form of what? Chain. You say chain spherical bacteria. Now, this chain spherical bacteria are called what? Streptococci. Streptococci. Now we have different species of what? Streptococci. All right. We have Streptococci venezuelae. We have Streptococci erytheros, and we have Streptococci griseus. Okay. There are different species of what? Streptococci, and they are what? They occur in the form of what? Chain. You now have the spherical bacteria occur in cluster cluster spherical bacteria. Now this cluster spherical bacteria, they occur as what? Staphylococcus. All right, which means that Staphylococcus aureus is a cluster spherical bacteria. Staphylococcus aureus is a cluster spherical bacteria. Now you now have the rod-shaped bacteria. The rod-shaped bacteria, the bacteria that occur as what? Rod. You call them bacilli. Bacilli, okay? You call them what? Bacilli. Now, a good example of a bacilli organism is the Bacillus thermolevorans, and it is a bacterium that degrade naphthalene. So Bacillus thermolevorans is a bacterium that degrade what? Naphthalene. We'll get there. Now, the rod chain bacteria is called what? A streptobacilli. Please take note of that. You now also have some that occur in the form of what? Spira or spirillum bacteria. Okay, you now have some that you now have the vibro one, that is the vibro cholera. That is bacteria that cause what? Cholera. Okay, that is vibro cholera. It's a bacteria that causes what? Cholera. So these are the different shapes of bacteria and the organisms involved. Though as the class progresses, I'm going to be giving you examples of organisms having this shape. I know I've given us some, but more are yet to be given. Okay. Apart from what I just listed on the board now, you have other eubacteria. We have other eubacteria such as we have the red spherical bacteria. We have the red spherical bacteria. Don't forget, these are bacteria under eubacteria. That is, I said this subkingdom Eubacteria bionta. The division is the eubacteria. So these are the organisms under it. We have the red spherical bacteria. Good. We have the spiral comma-shaped bacteria. We have the spiral comma-shaped bacteria. Now we have the rickettsias and chlamydias. All right, we have the rickettsia and what? Chlamydias. The rickettsias are rod-like, while chlamydias are spherical. You now also have the spirochaetes. spirochatis. You also have the you also have a purple sulfur bacteria. We have the purple non-sulfur. We have the purple non-sulfur bacteria, and we have the green sulfur bacteria. Please don't say it's too difficult. Do not say that. Just keep playing the video over and over again, okay, for a better understanding. As you read your textbook, you can use this video to master it better. Now you have this red spherical bacteria, we have the spiral comma shaped bacteria, we have the rickettsias that are rod-like, and the chlamydias, they are spherical shaped. Now we have the spirochatis, we have the purple bacteria, we have the purple sulfur bacteria, we have the purple non-sulfur bacteria, we have the green sulfur bacteria. However, this purple sulfur bacteria may appear what? Red. May appear red brown. This purple sulfur bacteria may appear red brown because of the mixture of red, yellow, and brown pigments. Alright. Now these organisms, this purple sulfur bacteria may sometimes appear with red brown because of the mixture of red, yellow, and brown pigments. So please, another example of Eubacterium is the cyanobacteria. The cyanobacteria is also a good example of what? Eubacterium. Now you also have you also have the chlorosy bacteria. Please, this chlorosy, this is like chlorosy bacteria. Now these chlorosy bacteria have two components. We have one, diprochlorone, and diplochlorophthalate. The cyanobacteria are under Eubacteria. The chlorosy bacteria are under Eubacteria. Now these chlorosy bacteria have two components, the prochlorone and the prochlorothrix. They are the components found in the chlorosy bacteria. You know, bacterium is singular, then bacteria is plural. It doesn't say bacterias. All right, it is what? Wrong. Now, so these are the organisms you have under Eubacterial biota as what? A subkingdom, and they are known as what? The true bacteria. Now, these true bacteria, they have a cell wall, a special type of cell wall called peptidoglycan. Okay, before I do that, let me tell you what a scientist did. I'll quickly do this by going into that. Now, Christian Brown, in year 1884. Now, he was my student. Yes, I taught him. He was my student. Now, in year 1884, based on the based on the nature or the properties of the cell wall, he did a particular experiment. This was what he did. Now, he took a smear. He took a smear of what? Bacteria, okay? Now, smear is a substance that can be spread or rubbed on another what? Surface. Now, he took a smear of bacteria, okay? He soaked this smear with violet dye. Listen, oh, he took a smear of bacteria, he soaked it with what? Violet dye. Then he treated it with iodine. He treated it with what? Iodine. He washed it with alcohol, and he counterstained it with, that is counterstained. He counterstained it with sophroniene, which means that the stain that was used by Christian Graham was sophroniene. The washing agent was alcohol, while the substance he used in treating the smell was iodine. Then the one he used to soak it or as a mordant was what? Violet dye. Excuse me. As soon as he conducted his experiment, at the end of his experiment, he observed something. And what did he observe? He observed that some of the bacteria, that is, he took a smell of bacteria. This smell now represents what? Bacteria. Now, he observed that some of the bacteria now appeared blue to purple. Some of the bacteria appeared blue to purple upon taking up this what? The violet dye. Some of the bacteria took up this violet dye and appeared blue to purple, and he called these bacteria the positive gram bacteria. After doing this, at the end of his experiments, he observed that some bacteria took up this violet dye and appeared blue purple, and he called this bacteria positive gram bacteria. Then some of these bacteria were washed. That is, their violet dye was washed by this alcohol, and they appeared pink to red. We call these guys negative gram bacteria. This was his experiment. Violet dye was used to soak it. Then the iodine was used to treat it. Then alcohol was used to wash it. Then sophroniene was used to what? Stain it. Okay, violet dye cannot act as a mordant. So please, it is what? This iodine, please, not violet dye. Okay. Now, after doing this, some bacteria took up this violet dye and appeared blue purple. We call them positive gram bacteria. Then some of these bacteria were washed. That is, their violet dye was washed away by this alcohol, and they appeared pink red, and we call them negative gram bacteria. In conclusion, the experiment conducted by Christian Brown is now used to classify bacteria based on their ability to take up what? Stain gram. Now, this experiment he conducted was then used to classify different type of bacteria based on their ability to take up the gram stain or stain gram, as the case may be. Now, this was Christian Graham's word experiment. Please take note of these substances that were used. Now, after doing this, the cell wall of some bacteria, I told you that the Archaeobacterial biota as a subkingdom, the Archaeobacteria do not have peptidoglycan. They do not have it. But the eubacteria, they have what? Peptidoglycan. Now, after his experiment, he noticed that he then concluded by saying that the one having a lot of peptidoglycan, he then concluded that the set of bacteria having a lot of this peptidoglycan, we call them the positive gram bacteria, while the one having less peptidoglycans were called negative gram bacteria. Now, please, what is a peptidoglycan? A peptidoglycan is the cell wall of bacteria that is made up of interconnected amino acids and polymer of carbohydrates combined with a muramic acid. I said the peptidoglycan is a cell wall of bacteria that is made up of amino acids, polymer of carbohydrates, and what? Muramic acid. They are all what? Interconnected. This description is restricted to what? The peptidoglycan. However, outside externally to the peptidoglycan is the glycocalyx. Externally to the peptidoglycan is the glycocalyx. This glycocalyx is made up of three entities. It's made up of the glycolipids. It's made up of the glycoproteins and the proteoglycans. Right, these are the components of the glycocalyx. So glycolipids, glycoproteins, and proteoglycans are the constituent or the component of what? Glycocalyx. I see that glycocalyx is found externally to the peptidoglycan. Please, I forgot something. Thank God I just remembered. Okay. Now the eubacteria, they have a slimy sheath. All right, that gives them the ability to stick to their host. They have a slimy sheath. Now, please, the type, okay, some slimy sheath could be very weak, and you call them what? Loose slimy layer. Now, this loose slimy layer can be washed away easily, but the one that cannot be washed away easily is called a capsule. Which means the capsule is a dense and thick slimy layer found in certain bacteria that it uses to stick itself to the host. But as for the loose slimy layer, it is easily washed away. It is washed away easily because it is not loose. It is not what? Dense. So please take note of that. Please, when bacteria stick to themselves, when two or more bacteria come together, they stick themselves, they form what is called a biofilm. And it is this process that actually results in the capsule. Alright, please. The capsule is that slimy sheath, is that thick and dense slimy sheath that is found in bacteria that gives them the ability to stick to the host in which they are found. So please take note of that. Now, okay, I've given you the experiment of the experiment of Christian Brown in year 1884. Okay, now it categorized some bacteria as positive gram bacteria and some as what? Negative gram bacteria. Now, what are the negative gram bacteria now? These markers are becoming bad already. The negative gram. What are some examples of these negative gram bacteria? We have the Treponema. We have Treponema pallidum. Treponema pallidum causes what? Syphilis. All right. A syphilis is a disease that is contracted during sexual intercourse. So you can say it's an example of what? STD. Another example of negative gram bacteria is the Neisseria gonorrhea. The Neisseria gonorrhea is the Neisseria gonorrhea, which causes gonorrhea, Treponema pallidum, which causes syphilis. The both of them are bacteria that are associated with sex. So which means gonorrhea and syphilis are STDs. Okay. Another example of negative gram bacteria is the Escherichia coli. All right. Escherichia coli is also what? A good example of negative gram bacteria. Then the last one for now is the cyanobacteria. The cyanobacteria is photosynthetic. I think I said something about it before. It is what? Photosynthetic. It has it has blue pigment called what? Phycocyanin, and it has a red pigment called phytoerythrin. Okay. Now these are examples of what? Negative gram bacteria. Now the next one is what? Let us check some examples of what? The positive gram bacteria. The positive gram bacteria. Now examples of these bacteria are one, the actinomycetes. The actinomycetes. Two, we have the Clostridium tetani. We have the Clostridium tetani. And three, we have the Mycobacterium we have the tuberculum and the Mycobacterium tuberculosis. All of these are examples of what? Gram positive bacteria or positive gram bacteria. So please take note of that. Now I've given you examples of negative and positive gram bacteria. Now our question will be, who was the first to discover this bacteria that we'll be discussing? Who was the first scientist to discover this bacteria? The first scientist that discovered the bacteria is called Anthony von Leeuwenhoek. All right, Anthony von Leeuwenhoek. Anthony von Leeuwenhoek was the first scientist to discover the bacteria in 1832 to 1823. All right, is it? Yeah, 1822, then 1923. Sorry, this 16 now 18. Yeah, then Robert Brown. We have 1822 to 1873. Okay, 1822 to 1895. Yeah, 95. Now is it Robert Brown now? No, not Robert. Sorry, Robert Brown is the person that's actually that was the first person to describe what the nucleus is. It just entered my head. It's not it. I wanted to write Louis Pasteur. Alright, Louis Pasteur was the person that first introduced the study of bacteria called bacteriology. Now, Anthony von Leeuwenhoek discovered the what's it called now? Discovered bacteria. The study of bacteria, which is bacteriology, was discovered by Louis Pasteur. Take note of that. Now, another scientist called Robert Koch. Robert Koch was a person that discovered the various diseases caused by microorganisms. He was the first person to discover the various diseases that are caused by microorganisms, and he gave some rules regarding how these microorganisms cause such diseases. Okay, Louis Pasteur is to bacteriology, Anthony von Leeuwenhoek is to bacteria, then Robert Hooke is to the various diseases caused by bacteria. Please, tuberculosis was discovered by Robert Koch in year 1882. Robert Koch in year 1882 discovered tuberculosis. Like the causative agent of tuberculosis is called what? Mycobacterium tuberculosis. So please take note of that. But I'm still saying it. Bacteriology, Louis Pasteur; bacteria, Anthony von Leeuwenhoek; Robert Koch, the various diseases caused by bacteria. Then in 1832, he discovered a causative agent of tuberculosis, which is what? Mycobacterium tuberculosis. Hope it's recording. Yeah, it's recording. Thank God. Okay. Now I've given you all these idiots. All these things I've given you, they are all important. So long PBB 111 is concerned. Okay, they are all important. Now the next thing we are going to be treating is the pathogenic forms of bacteria, the beneficial form of bacteria, and the medical importance of what? Bacteria. Now, we have, please, take note of this. Plants pathogenic bacteria are dash shape. Plants pathogenic bacteria are dash shaped. Let me add D to it. A, rod shaped. B, spherical shaped. C, chain spherical. D, cluster spherical shaped. So what is the answer? Please, plants pathogenic bacteria are rod shaped. Take note of that. They are usually what? Rod shaped. Plant pathogenic bacteria are what? Rod shaped. They are rod like in shape. Now, what are these? A good example is a particular organism that causes the growth of, or the massive growth of trunk trees. We call this word crown gall. Crown gall. Now, the bacterium that causes the massive growth of trunk trees is called the agrobacterium. It's called agrobacterium tumefacens or tumefacens. So please, agrobacterium tumefacens are bacteria that are responsible for the massive growth of trunk trees called what? Grow gall. Take note of that. Now, we also have bacteria that are beneficial. We have some bacteria that are what? Beneficial. These examples are good. These examples of beneficial bacteria are, we have one, the rhizobium japonicum. We have the Rhizobium japonicum. I think we also have the uh, Clostridium pasteurianum. All of these are beneficial bacteria. Okay, but this one is a pathogenic what? Bacteria. Now number three, you now also have the Azotobacter indicum. You have the Azotobacter indicum. Then number four, you also have the denitrifying bacterium called Clostridium denitrificans. All right, so Clostridium denitrificans is a denitrifying bacteria that is what? Beneficial. Azotobacter indicum, Clostridium pasteurianum, then Rhizobium japonicum are all what? Beneficial bacteria. Which of these is not a beneficial bacteria? They'll give you this and add this other one, so this becomes the answer. Now we also have some bacteria that are used as bioremediations to toxic waste. All right, we have some bacteria that are used as what? Bioremediation to toxic waste, that is pollution. Such bacteria are one, we have the we have the Bacillus thermolevorans. The Bacillus thermolevorans degrade naphthalene. Then number two, you also have the Pseudomonas. You have Pseudomonas grisosus. These bacteria, they degrade normal hexadecane. All right, these Pseudomonas grisosus, they degrade what? Normal hexadecagon, or hexadecane. That is what? The 16th member of what? The arcane series. And number three, you also have the Pseudomonas putida. All right, they degrade trichloroethylene. All right, do not say these names are difficult. No, they are not. Okay, just read. Keep reading your textbook, and you can use this video to also follow up. I said Bacillus thermolevorans degrade naphthalene. Then Pseudomonas grisosus degrade normal hexadecane. Then Pseudomonas putida degrades trichloroethylene. So please take note of that. Now we also have some bacteria that are medically important. They are medically what? Important. But before I take that, let me introduce something. Let me introduce something here. Let me introduce the actinomycetes. Now, these actinomycetes, they appear mold-like. They are mold-like what? Organisms. They are mold-like organisms, and they look like the true fungi molds. All right. Which of these set of bacteria are mold-like, hence looking like the true fungal molds. Please, it is what? The actinomycetes. Now, these actinomycetes, they lack nuclear membrane. They lack nuclear membrane, and they do not have a membrane bound organelles. Please, these features I'm giving are restricted to just actinomycetes. The actinomycetes are mod-like in shape. They lack nuclear membrane. They do not have membrane bound organelles, okay? And they are mostly anaerobes and saprophytes. They are mostly anaerobes and what? Saprophytes. And they have the ability to bring about what? Decomposition. They have the ability to decompose organic what? Matters, thereby releasing what? Nutrients for the growth and good development of what? Plants. Now, we have different species of actinomycetes. For example, now we have the actinomycetes israeli. We have the actinomycetes israeli. Now, this actinomycetes israeli causes oral cavity what? Infection. Oral cavity infection. All right. This actinomycetes israeli causes what? Oral cavity infection, which means this idiot is what? Pathogenic. Now, you also have some actinomycetes that are medically important. They are medically important. They are the streptomyces. All right. Examples of these medically important streptomyces are we have the streptomyces venezuelae. Now, this streptomyces venezuelae produces chloramphenicol. All right. This streptomyces venezuelae produces what? Chloramphenicol. Okay, phenocol. Now, this chloramphenicol is an antibiotic. It's an antibiotic that fights against intestinal infection. All right. The fight against what? Intestinal infection. It is an antibiotic produced by the streptomyces venezuelae. Please, the function of this chloramphenicol is to fight against what? Intestinal infection. Another species of the streptomyces is the streptomyces erythrox. Now, this streptomyces erythrox produces an antibiotic that is called what? Erythromycin. Streptomyces erythrox, which is a type of bacterium that produces an antibiotic called erythromycin. Now, the function of this erythromycin is to fight against whooping cough. All right, so you can see now that they are medically important. You now also have another type of bacterium called Streptomyces Is it Grisessus now? Yes, Streptomyces receptors, it produces what is called streptomycin. Strepto, streptomycin. And this streptomycin is what? An antibiotic. An anti what? Biotic. So please take note of that. Take note of that. Okay, is there another one that I'm yet to give? So please, these, and these, they are medically important because they produce antibiotics that are very important. And this actinomyces is really causes what? Oral cavity infection. So take note of that. Take note of that. Now, these are all you need regarding or anything that has to do with actinomyces has been dropped here. Now, please, I would have given us this earlier. It just entered into my brain now. Now, I gave us that we have different species of bacteria. I said we have the rickettsias, we have the chlamydias, and we have the sporochetes, we have the red shaped, that is red spherical bacteria, and we have the spiral comma shaped bacteria, we have the cyanobacteria, and I said we also have the chlorosy bacteria that's comprise the prochloron and the prochloratrix. Okay, now all of these mentioned bacteria, there are two bacteria that are different from every other microbacteria, from other bacteria to be specific. Now these two examples of such bacteria are, one, they are the rickettsias. They are the rickettsias and the chlamydias. The rickettsias and the chlamydias have different characteristics, or characteristics that distinguish them from every other what? Bacteria. Let us do this. Now, these two bacteria were once considered as what? Viruses. They were once called viruses because they occur in the intercellular space of eukaryotic cells. Which of these bacteria were once called viruses? Please, the rickettsias and the chlamydias were called viruses because they occurred as intercellular parasites, intercellular what? Parasites of eukaryotic cells. Now, what are the features that they have? One, I told you that normal bacteria will not have DNA and RNA at the same time. It's either you have DNA, it's either the bacterium possesses DNA or what? RNA. But these idiots, these bacteria, they have DNA. It never do for them. They have RNA. No. They have ribosome. You can see now that they are quite different. They have DNA, they have RNA, they have ribosome, and they have the ability to synthesize their own nucleic acid protein. You can see that they have DNA, RNA, ribosome, and they also have the ability to synthesize their own nucleic acid protein. That is not just it. They also have enzymes. They have enzymes that make them to carry their metabolic activities. These guys, they are very peculiar. They are special. They are very, very special because these features are absent in other other bacteria. And again, apart from that, it's just that they depend on the host for what? ATP, which means that they cannot generate their own energy. They depend on the host for what? ATP, called adenosine triphosphate. Okay, so these are the features of what? The rickettsias. DNA, RNA, ribosomes, nucleic acid are present. They have enzymes that make them to carry their metabolic activity, and they are dependent on the host for what? ATP. Now, since we've established that fact, let me give you some examples of organisms of this that cause different diseases. For example, now excuse me, let me rub off this. Now, oh good, I don't like this. It's not bright. It's not bright again. Now, the chlamydia, the chlamydia causes a disease called, okay, let me use the word psittacy. Yeah, chlamydia psittacy. Is it S? Okay, it should be S. Chlamydia psittacy causes a disease called trachoma. This trachoma has the ability to cause what? Blindness by evading the eye. Okay, please, the chlamydia psittacy causes what? Trachoma, a disease that leads to what? Blindness. Now, this rickettsia, the rickettsia prowazeki causes typhus fever. I did not say typhoid fever. Typhus fever is caused by Rickettsia prowazeki. Now, typhoid fever is caused by a bacterium called what? Salmonella typhi. Salmonella typhi causes what? Typhoid what? Fever. It causes typhoid fever. Take note of that. Which one again? Okay, now yellow fever is caused by flavivirus. Okay, it's caused by flavivirus, and the vector of this yellow fever is the Aedes mosquito, which is called what? Haemophilus. Haemophilus species is a vector of what? Yellow fever. Flavivirus causes yellow fever. Salmonella typhi causes what? Typhoid fever, and it is what? Water and food borne. This typhoid fever is a water food borne disease. The Rickettsia prowazeki causes what? Typhus fever. Then trichoma psittacy causes what? Trichoma. Please take note of that. Oh, okay. I think something just all right, let us do this. Now, I explained something when I was giving you gram-positive bacteria and gram-negative bacteria. Now, there are two species of microbacterium. There are two species of what? Okay, microbacteria. We have the microbacterium. Oh God. We have the mycobacterium tuberculosis. Now, this is that of bacteria that causes tuberculosis, which is quite understandable. We now also have another called Mycobacterium leprae. I know you already know what it means. Mycobacterium leprae is that bacterium that causes what? Leprosy. It causes what? Leprosy. Leprosy. Okay, so please take note of that. Now, based on the properties of the cell wall, based on the properties of what? Cell wall. Now, there's what we call mycoplasmas or pleuropneumonia like organisms. All right, mycoplasmas or pneumonia-like organisms, which is PPLO. All right, they are this. I think I said something like that before. They are the smallest organisms on Earth. They are smaller than even the bacteria. They are smaller than bacteria. Take note of that. And these idiots, these wicked type of bacteria, they are found in the phloem of what host plant, thereby interrupting the movement of food through translocation. Now, mycoplasmas or pleuropneumonia-like organisms, that is PPLO, are found in the phloem of a host plant. What is their aim? Their aim is to interrupt the movement of food through what? Translocation. You know the phloem carries manufactured food through a process called what? Translocation into other parts. Now, when the medium through which the food molecules are actually transmitted is blocked, what do you expect? The plant will start to what? Die out. Okay. Now, what is now the impact of such a thing? Now, it leads to coiling of leaves, to stunted growth, sure, slow growth, and leads to yellowing of leaves. Yeah, yellowing of leaves, because food materials are no longer what? Translocated into these parts. A good example of a mycoplasma is called mycoplasma. Oh god, I'm almost rounding off. A good example of such uh, mycoplasma or pleuropneumonia-like organism is called what? Mycoplasma gallisepticum. So Mycoplasma gallisepticum has the ability to cause diseases. These are abnormalities. Where are they found? They are found in the phloem of host plant, in order to interrupt the movement of what? Food through translocation. So cyanobacteria, I've treated the actinomycetes. The next one we are going to be discussing is what? The cyanobacteria. I don't know if I should take it or I should just leave it for now. Okay, let me make introduction to cyanobacteria. Now, cyanobacteria are the only type of bacteria that have the ability to fix nitrogen and release what? Oxygen through photosynthesis. Now, they're able to release oxygen because they carry out what is called photolysis of water, or photochemical reaction of what? Water. They split water molecules into hydrogen ion and hydroxyl ion. From that water, oxygen is what? Liberated. Now, I said they have the ability to release nitrogen, okay, fix nitrogen to the soil and also release what? Oxygen. So they are only the organisms that do that, okay? And these organisms, they have two pigments. The blue pigment called phycocyanin, the red pigment called phycoerythrin or phycobilin. Now, there are different species of cyanobacteria. Now, normally cyanobacteria are non-motile, they do not move, but there are some that move through by the lashing movements of their filaments. All right, there are some that move by the lashing movements of their filaments. A good example is the Glucapa cortinata. All right. Okay. Now, this Glucapa cortinata are actually a type of cyanobacterium that occur in the form of, they occur as a single chain. All right, single what? Chain. So they occur as what? Single chain. There are some that occur as colonies. There are some that occur as colonies. They are called Anna, Annabella uh, Azuna or Azula. Yeah. This Annabella Azula. Okay, is it Annabella? Annabella. Oh, good. Annabella Azula. They are filamentous cyanide bacteria. Okay, they occur as what? Colony. Now there are two filamentous examples of cyanobacteria. They are the nostoc species and the Annabella azula. They are the both of them are what? Filamentous. Now I said that cyanobacteria do not move, they are non-motor, but some have the ability to move by the lashing movement of what? Their filament. Okay, so a good example is, I think I did not give example before. Oscillatoria species. Oscillatoria species is the motile bacteria that move by the lashing movement of what? Their filaments. While some cells of some organisms could just occur as one, as a single strand. A good example is the Glocarpa coordinata, okay? But the one that occur as what? Colony. Their cells are not joined together, they now occur as what? Colony. And they are filamentous. Example is what? Annabella azula. Another example of filamentous organism is what? The nostoc species. Nostoc species. So which means that the nostoc species and the Annabella azola are the only filamentous what? Cyanobacteria. So at this point, we call it a day. In my next class, I'm going to be telling you everything you ought to know about what? Viruses. So at this point, we call it a day. Thanks for watching and endeavor to subscribe, like, share, and comment, please.